All right. So my name is Rabern Simmons. I am the curator of fungi at the Purdue University Herbaria. I will be talking. Oh, okay. I will be talking about why it is we call it the herbaria and not the herbarium a little bit later on. And my title is a reference to the story of Rumpelstiltskin. That hopefully the gold and black makes sense when I talk about Purdue. And I will get into why it is we talk about a Haymow or a Haymow later on in discussion as well. But the first question that I ask, and I think there's actually people here that actually know the answer to this, but just what is an herbarium? And for the most part, you can think of an herbarium, rest on my slide, but not here. Let's. There we go. Okay. So what is an herbarium? And you can really think of it as a collection of dried and preserved plants with a little p, because a lot of things that have historically been thought of as plants are not now classified as true plant life. Those can include algae, which themselves are just a basket organism. Fungi, there's fungi with a big F and fungi with a little f. And these herbaria have been around for quite some time, popularized. We were talking actually about curiosity cabinets early on, and how that kind of led to the creation of her formal herbaria instead of just private collections. And in the case that I'll be discussing later on, an herbarium that is exclusively fungi, is exclusively a collection of fungi, is often called a fungarium. The roles of these herbaria are multifaceted, but really you can think of it as preserving a snapshot in time. You have a specific collection from a specific place at a specific time, even of year, not just in history. And a lot of the specimens that have been collected can no longer be collected in certain areas. There's actually next week, Chad Evans, our local meteorologist, will be doing a presentation for the botany and plant pathology department because he used to do the Tree of the Week's stories, and he has a passion for these plants of the southern part of the state. And there's so many things that he's already just in passing talked to me about that don't exist anymore. These terrains that don't exist. So the herbaria serve as a snapshot of those. It also serves as a place to put the voucher specimens for new species. New species are still commonly being described specifically for plants, and very much so for fungi, because there's so few people that are interested in these organisms, and plants just because there's a lot more people that are looking for these things and how the species is truly defined. So we have these type specimens upon which a new species was described. One of the people in the Indiana Academy of Sciences like to refer to herbaria as not only libraries, but a place where you can store forensics evidence to be used for future investigations. And these can, again, span out larger than just botany into agriculture, conservation, epidemiology, plant management, and now our understanding of climate change. And finally, the part that I think maybe I enjoy the most is just getting people interested in botany, fungi, mycology, and just all these interactions and symbioses in the natural world. So I'll be going over that as well. We'll take a moment now to say that we will be open for Springfest. If you want to stop by the herbaria, we are at Site 11, I believe, from the meeting that we had earlier today. And I have some propaganda of flyers and brochures, and also some little mushroom Purdue University botany and plant pathology stress relievers. So feel free to take some of those. Feel free to take some of those when you leave. So the Purdue University Herbarium has always existed conceptually since the formation of Purdue University. John Hussey officially started the collection in 1874. Within the first 5 years of his sampling efforts and also receiving specimens from around the world, the collection had grown to about 2,000 specimens. And he was had to take retirement in 1880. The next person that was kind of in charge of this collection was Charles Barnes. Charles Barnes was a high school teacher at Lafayette and is an Indiana native, but he's perhaps most widely known for his work at the University of Wisconsin, where he coined the term photosynthesis. So you can kind of think of Purdue as the home of photosynthesis. But while he was here at Purdue, he expanded the herbarium to a total of 7,000 specimens stored traditionally in the basement of buildings, and at that point stored in the basement of the Department of Biology.
Yeah, dark places, I understand. I was wondering, the Reverend Hussey, his medical retirement, did that have anything to do with his collecting of spores and fungi?
I don't think so. I think it was unrelated to that. I can't recall the specific reasoning behind his medical retirement. Stanley Coulter, of whom Coulter Hall was named, and at one point the herbarium was renamed in his honor, the Coulter Herbarium, or when it was officially named something other than just a Purdue University Herbarium in general, was the next person to be in charge of the collection. And when the Coulter Hall was built, that's where the collection also went, also in the basement. The next person that is most readily associated with the collection is Ralph Kriebel. Kriebel was a high school teacher in Kinland and worked on a lot of stone carving and also wood carving. He had a meeting, or he met Charles Deane in the 1930s, and Deane was the first state forester of Indiana. He literally wrote the book on the flora of Indiana in 1940, in which he had traveled to over 1,000 townships within the state, specifically looking at ferns and oaks. He really had a passion for ferns and oaks. This passion was translated on to Kriebel, who also collected a lot of ferns and oaks. And Deane is also known as the namesake for the Deane Oak Monument Forest, which is the smallest state preserve in Indiana at 0.2 acres. He purchased this land in 1915, I believe, for $75 because there were these, I can't remember, there's 2 trees or 3 trees that really interest him, these 3 oaks, and he thought they were a new species. He sent them to the Missouri Herbarium for collection verification. The people there named the new species after him, so it's Quercus demii, and it was eventually discovered that this is probably just a hybrid species of 2 other things, but it is still, when he purchased the land, he did so because the road there, I assume one of those roads there was going to be widened, and the land was just going to be decimated, trees destroyed, and he really thought these were very interesting oaks. He followed them for about 15 years before he purchased the land, and he immediately donated it to the state. So again, this is the smallest state preserve in Indiana. It is the Deane Oak Monument Forest. So according to the state of Indiana, a forest is constituted of no fewer than 2 to 3 trees, I suppose. So Kriebel met Deane. Kriebel was also working for the U.S. Soil Conservation Service as an extension specialist here at Purdue, starting in 1936, and that's when he started to really focus on his contributions to the Purdue collection. Yeah.
That Deane oak, it is inhabited by Gravola frondosa.
Oh yeah, you told me that.
Did you see I collected a specimen, it's in the herbarium.
Yeah.
So maybe you see.
So maybe those oaks might not be there.
Well, yeah, it's doomed,
Yeah.
actually, but.
It has a fungal infection.
It's a fungus that inhabits oak trees. It's delicious. It's one of the best wild mushrooms to eat. It's called hen of the woods.
Deane also did send some, there are 2 individuals, I believe, at the Harvard Arboretum of these trees as well. But it is no longer recognized as a valid species.
Yeah, it's a hybrid.
Yeah.
It's still.
Yeah.
And there are 3 progeny trees in that block.
Okay, I can remember how many.
From acorns that came from that.
So Kriebel started working officially for Purdue in 1943. He unfortunately passed away in 1946, just three years later. And he had at that point accrued 10,866 mounted specimens, which were referred to by his mother as his Haymow or his Haymow. And his mother did not want these in her house anymore upon his death. And she sold them to Purdue University. At the time of that contribution to Purdue, this was recognized as the largest collection of Indiana plants by any one person, except of course the work that was accomplished by Dean. The Coulter Herbarium was rededicated in 1961 to be the Kriebel Herbarium, which is what we now still continue to call it. All herbaria have specific letters associated with them for an abbreviation, and the abbreviation of the Kriebel collection, is the PUL. You might know the name Kriebel. Ralph Kriebel had one son. His name was Robert Kriebel, and he has been associated with the Purdue University Press and written, among other things, a biography about Charles Dean, plain old Charlie Dean. And just this past year, some of the Kriebel descendants came to visit the collection.
I met Bob Kriebel while I was working there, or was in an email conversation. He was very nice about answering questions about the herbaria.
Yeah.
And
it was really very helpful for them to come in and actually kind of look at some of these specimens, and they were able to read some of the handwriting that I was having trouble with, and they were actually saying like, actually, I don't think that's Ralph's handwriting. I think that was his wife's handwriting. So it's very interesting reading to have.
Are you gonna tell us a little bit about where Dean's collection is?
I believe Dean's collection is mostly at IU.
Yes, it is.
And then the specifics behind it, I'm not gonna go into.
You're not
but
convinced?
I'm not convinced.
Oh,
I-
well, there's a
Good
story
story.
behind that.
Okay, I'm not, I just know he wasn't that great a fan of Purdue.
I could
Yeah.
tell a story, but
Well,
I got too
I'll
many
do it.
stories.
I'll do it.
Okay.
Let me do it just a little
Okay.
faster, okay? It went into a it had to be moved because they were building something where it was located at that time. And it turned out that they moved it into the men's gymnasium.
Right.
Okay, so you heard.
Yeah,
that.
for the.
And the students who were in there were showers and they were using the sheets as
Floor
foot
mats.
pads,
Yeah.
to, keep their feet dry as they move from one place to another. And Dean saw that.
Yeah.
And said,
I'm not,
The heck with
gonna
you.
produce my stuff.
yeah, he took all this stuff and gave it to IU.
Yeah, we do have some Dean collections,
So you
but
know
not.
that story then.
Yeah, yeah.
But you just don't believe it.
I don't have the proof. I've
Oh, no, I know.
never
That's-
seen
it's-
the footprints on the.
Yeah,
the
I
specimens.
never saw any specimen that had footprints.
Yeah.
No, well, have you looked at all of Dean's specimens?
No.
Good
Well,
handful of them.
the strange thing about, because I had heard the story about Dean pissed off at Purdue and wouldn't let him, but then like 2 days, within the first week of me working there, I found, this Charles Dean specimen.
Oh, yeah.
There are Charles.
We do have Dean specimens,
Oh,
definitely.
it would probably reach
Yeah.
2,000. I don't
So
know.
especially that demo, we have multiple decades actually of the demo collection. So we definitely have our
Yeah.
deep stuff as well. Concurrently though, to that whole story that I previously talked about the Purdue University Herbaria, there was also Joseph Arthur. Joseph Arthur was the first graduate of Iowa State College in 1872. He was the first recipient of a doctoral degree from Cornell University. And it's not just because his name started with an A, his last name started with an A. He was just the first person to do so. And he started his own personal collection in 1882, though he did a lot of work before that as well, including winning some awards at a couple of World's Fairs, or at least one World's Fair before that. And he was employed by Purdue in 1887, so 5 years after he started this collection. And he was the head of the department of what we now have as the Department of Botany and Plant Pathology. He worked at the Agricultural Research Station. He's a charter member of the Botanical Society of America, the American Phytopathological Society, the Indiana Academy of Science, the Mycological Society of America. And he's the only person that I'm aware of that has been the president of the Botanical Society of America twice. Once in 1901, so he was 51 years of age then, and then again in 1919, so he would have been 69 at that point. He also wrote, oh, and I meant to have this pulled up already, but I didn't have the opportunity, I'm sorry. But you can go to our website and listen to the song Viva Purdue with words by Evelyn Stein, written the music by J.C. Arthur. That was in 1901, I believe. So he really did enjoy his time at Purdue, and even did a lot of things in his later life. Staying here in Indiana, still lived in Lafayette for the majority of his life, and just really loved Purdue. However, after retiring in 1915, he continued to come to campus to visit his collection that he started in 1882. That he brought to Purdue in 1887, that Purdue really never gave him money to help continue, that he did all this stuff from his own personal funds, his own personal time. After 3 years of that coming and going to visit his collection, he kind of got tired of it. And in 1918, in the middle of the night, July 4th, he rented some moving vans. He went on campus, he loaded everything up, and he took his ball and he went home. He brought it back to his house on Columbia and 10th, and just had his specimens there. Purdue was not pleased with this arrangement. They contacted the Tippecanoe County Sheriff's Office, and in August of 1918, J.C. Arthur and Purdue University Board of Trustees reached an agreement. There were three major points. The first being, one, Arthur could come and go as he pleased and use whatever resources he wanted. He was the charter member of all the societies. He was a very prominent mycologist, botanist, epidemiologist. People knew him, and Purdue didn't want to be seen as the people kicking this old guy off campus. Yes.
Back up to his name, because his name didn't mean anything until you got to this slide. What was his name, please?
Joseph C. Arthur. Or often J.C. Arthur. The only person referred to him as anything other than Joseph was his wife. And I think she could get away with calling him Joe.
Joe, get those mushrooms on it.
So Arthur, he had those three major points. First being he'd come and go, do whatever he wanted. Second was that Purdue University would pay him $1,000 in 1918 money to essentially buy the collection back from him. With this money, he took his wife on a tour of Europe. Later in the 1930s, Purdue University bought his reprint collection because we couldn't just download a whole bunch of PDFs of other people's works back in the 1930s. And they purchased it for $450, and he took his wife on another tour of Europe. The third point was that he wanted his collection to be kept separate from what at that point was just referred to as the Purdue University Herbarium. He didn't want his name to be forgotten, his work to be just overwhelmed by what at that point was the larger of the two collections. He didn't really have anything to fear. His collection is the largest of the two collections now, but as we now have the Arthur Fungarium, because it is exclusively of fungi, the Arthur Fungarium is exclusively rust fungi on obligate plant hosts. And that's because Arthur literally wrote the book on rust fungi. And rust fungi are parasites on wild plants, agricultural plants. If you know about fungal diseases, you may have heard about rust fungi. And historically, since Arthur, there have been 4 full directors of the Arthur collection, and they have all been specialists on rust fungi. He did a lot of work, a lot of papers and books on the subject as well. and passed away in Brook in 1942. He is buried in Springvale Cemetery, next to Emily Stiles Potter, who was his wife that had previously passed away from appendicitis in 1935. And this is the house that she grew up in, that they both shared, and they had no children.
And just for the record, for those that know, that's the house that William Potter lived in before they moved to the Potter Mansion on top of 9th Street.
Yeah, I've told people like, oh, they lived in the Potter house. They're like, oh, they lived in the Potter house. Like,
The
no.
first Potter
Oh,
house.
yeah, different one.
He retired in 1915, but the manual, the Rust, was published at 19 years, was it 19?
Oh, it was posted in
19 years
1934.
later.
Yeah.
Was he working on that all the time? Well, he was retired from Purdue, but like you said, he visited Herbaria.
Yeah, he was.
Actively working on that, and it took him that long to get it together.
Yeah, he was definitely working on the I mean, the manual of the Rust of the United States and Canada is his magnum opus. He spent the majority of his life working on it.
Yeah.
And I'll go into how the people that helped him do that in a couple of slides more. This past year, though, we did celebrate the 175th birthday of J.C. Arthur. One of his contemporaries said that the life and work of Dr. Arthur illustrate the attainments which may be achieved through real resistance to discouragement, industrious habits, sound scholarship, unflagging persistence, and high purpose. And his protege, not his student, but his protege, George Cummins, who was the second director of the Arthur Collection, stated that J.C. Arthur was a remarkable man. He decided early on a career in botany and proceeded with unremitting persistence to reach that goal. By any standard, he succeeded. And you can really tell that these people just really had a passion for Arthur and his great accomplishments and works. You can go again with the flyers that I have and on to our Herbaria website to read a full biography that I've written about J.C. Arthur that goes into more detail than I'm talking about here. And that can be said for most of these topics I'm talking about tonight, that you can find a lot more information on our website. Yeah.
Maybe you'll talk about it, but soon after I started working there, I pulled off, there was a publication put out by Tony Swinehart.
Mhm.
And he put all this biographical stuff about J.C. Arthur. And one was his J.C. Arthur's autobiography. He wrote it himself about
Yeah.
his career. And in it he mentioned, and he's like bragging, right? I was the one who introduced the word ecology into American science. Have you
Okay.
seen that?
No, I didn't see that
It's-
particular quote.
There's the manual of the herbarium.
Yeah.
Tony Swinehart put it together.
Right.
It's a brown. it's
Yeah,
like this.
I know.
Okay, you know,
Yeah, I
you'll
know.
find it in there.
Oh,
And
okay.
I'm thinking, I've never heard this.
I was gonna say the one thing I remember about J.C. Arthur I believe in that autobiography that he talks about is that his parents believed that he would not make enough money in botany
Oh, yeah.
to feed a cat.
Yeah,
That's
that's probably a lot of parents.
yeah.
Children going to go to botany.
So the next director was George Cummins. Cummins had a very interesting story on how he got to Purdue. He was originally a student at the University of Michigan, and he was a student of Calvin Kaufman. Kaufman trained another mycologist, E.B. Maines, Edwin Butterworth Maines. And E.B. Maines came to Purdue to be the mycologist on campus. When Arthur was still working on his book, he went to Maines and said that he would like to acquire somebody who had a fair amount of experience doing illustrations of fungi for his manuscript. So Maines reached out to Kaufman. Kaufman sent Cummins from the University of Michigan. And about a year later, Kaufman passed away. So Cummins was here expecting to eventually go back to finish his degrees with Kaufman. He then was here with Maines and thought, well, maybe I can finish my degree with Maines. But Maines instead went back to Michigan and took the role that Kaufman had previously had. So Cummins was just kind of floating in the ether here at Purdue and was working for Arthur, who at that point was in his 70s. Cummins, I believe, would sometimes refer to himself as Arthur's wet nurse. But he was kind of just here. Luckily, there were people in the department that took him in and helped him to actually finish his degrees here at Purdue. And upon completion of his PhD, he really just kind of rolled right into things and was a second director of the PUR starting in 1938. He was a full professor of botany by 1947, and he was the chair of botany from 1966 until I believe when he retired in 1971. He wasn't? Oh, I'm sorry. So he retired to Arizona, and he still continued to work on a lot of things while he was in Arizona. He was in 1981 the recipient of the Distinguished Mycologist Award by the Mycological Society of America. That is the highest honor upon which can be bestowed upon a member of the society. And he was in the inaugural class of that in 1981. I actually still have his plaque hanging over my office at my office desk. His last book was published at the age of 99, and he lived to 102. Arthur had previously lived until the age of 92 or 93, can't remember which at the moment, and Arthur lived to 102. Leading the MSA to kind of joke that if you wanted to live a long and prosperous life, that you could definitely do worse than studying rust fungi. But Cummins continued to be an excellent illustrator of the rust fungi, and we have so many specimens within the collection that have his hand drawings either on them or glued onto them. But sometimes just right there drawn onto the paper of the collection sheet itself. The herbaria have kind of floated around campus, various buildings, as I've already previously stated. But eventually in the 1980s, they were, or the 1950s, they were kind of reshuffled so that the Arthur collection was exclusively just the fungi, and the Kriebel was all the plants that didn't have fungal infections or all the other fungi. The PUR was transferred from biological sciences to botany and plant pathology in the 80s. And it was originally held, these two collections were held on the first floor of Lilly Hall. This is when they were redesigning it, and this is the final product. Anyone have any reasons why this wouldn't be the best space for an herbarium?
Much like.
Are those windows?
Those are windows. So that's facing what is now Mitch Daniels Boulevard. So you have weather and insects and humidity and just various things. When in the late 2000s, there was a call to insure the property. And as I understand it, the insurance companies said, no, thank you. So we had to relocate to now we are on the ground floor. We're not in the basement anymore. There's one floor below us. That's the basement. We're on the ground floor, but it is a sub ground floor. But we're on the ground floor of the of Lilly Hall. No windows. Sometimes I understand it's sunny outside. But it's a nice temperature controlled space where we have these things and keep them free of liquids and insects when most preventable.
Well, I heard that, well, I was there at that time when they moved it from the
Mhm.
ground floor to the to the ground floor. And the whole wing was being renovated.
Yeah.
Lilly Hall. So
Yeah.
it was like, that was, so they decided, and I remember there was this old guy. He was like an insurance specialist. He was like 80, 90 years old or something like that, but he was like this expert in like this kind of weird, and I didn't hear that they couldn't insure it or something like that, but.
Oh, okay.
That's, I mean, I'm not.
Yeah, no, that's how I've understood it.
That story, but I assumed that they had the money to renovate all that stuff, so put it downstairs.
No, it was definitely a need anyway.
It did expand the storage space though.
Yeah,
There were a lot.
with our compactors and everything.
But you know, one thing they had to get rid of all these old cabinets, which were just built for herbaria. And when I was there, I was trying to find homes for these orphan cabinets, because we had about 18 or so that that weren't designed, because they had to build some, the way they designed the ground floor, they had to install new cabinets. So I remember Sally Weeks took about dozen, maybe, I forget. I understand they're at the right.
There are some out at the right center that
Yeah,
seem
those are
very
hers.
similar, yeah.
First, she's at Wabash College. She's
There are some out at the right center that actually they've discussed giving back to us, or not the cabinets, but specimens out there that they had.
well, those were Sally's, yeah, right, that's, actually, when I was there, I
So
was
it's interesting that they want
was-
the stuff to
I
put
was
it back
trying
in
to
our
get Sally-
spaces. We've
Yeah.
given up space.
no, it's a good collection. She's really strong in salix.
Mhm.
You know, she was collecting a lot of that stuff. It's scientifically valuable.
Yeah,
I was trying to, in touch with NYBG to have a third, official third herbarium at Purdue and give it to her. And Barbara Thiers runs NYBG.
yeah,
And she, oh, no, we're not gonna put that in,
right.
But it really is good enough. It should be a official herbarium on its own. Of course, who's gonna pay to run it?
Right, yeah, we barely have the two. So the next director was Joseph Hennon. Joseph Hennon was a student of Cummins. And he did a lot of his own illustrations of rest fungal works, completing his master's and PhD with Cummins, eventually coming back to Purdue and himself becoming a professor of botany, taking over as director in 1971 and retiring in 1995. He just passed away this past year, again at the age of 96, again proving the point that rest fungi seem to have some life extending properties. Between 1995 and 2012, there was no formal director of the collection until the hiring of Kathy Aime in 2012. And since then, the collection has really kind of had this complete renaissance, and we've gone from 90,000 specimens to 125,000 specimens within the PUR. Kathy still does collections worldwide. She does a lot of traveling to Guyana. Just last year, they went to Mozambique, Iceland, a few other places. I know that there's somebody's going to Kenya next month. We have collaborations with people in Hawaii. Just that there's a lot of, she's done a lot of things on some very specific islands, Vanuatu and other places like that. So she's continued to be a dominant force in this mycological endeavor and was widely recognized for her contributions to mycology. Just this past year, she was the president of the Mycological Society of America, and she is currently the president-elect of the International Mycological Association.
Can I ask a
Oh, yeah,
question?
sure.
You mentioned all the travel, and I apologies, I'm totally ignorant on fungi and things. But why are they traveling to these places to collect the fungi when I would think they might be able to talk to experts in that country or that island or whatever, if they have.
I was going to say that is assuming that they do have
Yeah.
mycological experts, and
say, hey, look for this or look for that, and then just mail them in. Or is it a thing that when you find it, the location or the plant that it's on and everything is really just as important, just like archaeology?
Definitely, yeah. I think it does go back to just the paucity of mycologists in the world. I mean, the type of fungi that I specifically say that I'll go into a little bit more, there are probably 10 of us in the world that care. And there are probably 3 just in this country, and there probably were 5 about a decade ago, but they've either retired or passed away.
Yeah,
so-
over 100.
Yeah. So there's just not, mycology isn't a big draw. When you go out into the world, you see plants, you see trees, you see birds, you see mammals. You don't see fungi until they're a nuisance, or you see them for one day because it rained 3 weeks ago, and there's
Yeah,
just
and that's
this
when they
perfect
bloom,
confluence
yeah.
of events, and you see this reproductive structure, but that's it. You don't see the actual how it's doing things in the environment. So just historically, they haven't been that big of a draw. So yeah, there's that, just finding people that are interested, and these people here that have the experience that do go and travel around the world. In the case of Kenya, this coming trip, what they're currently trying to do is there's a fungus that infects coffee, and people really like coffee. They don't want there to be a rust that is killing coffee plants. And the theory is that somewhere, probably in Kenya, there is an alternate host. Rust fungi have a very complicated life cycle where the fungus is a parasite on one plant, then it sends a specific type of spore to another plant, and then produces a different type of spore on that plant, and it goes back to the original plant, and that's the simplest version. There can be up to 5 different spore types and different hosts, and those hosts are across families of the botany tree of life.
Now, I have some apple trees that got apple
Yeah.
rust, and I found out, oh, it comes from these, is it cedars?
Yeah,
Yeah, and it's like same thing, but it looks different
yeah.
on each plant.
And that's what Arthur really is, what he did with that manual of the rust, because he did almost 4,000 experiments by hand, like inoculating plants, to figure out, okay, species A on plant X is the same thing as species B on plant Y. It's just different parts of the life cycle. So he
Wow.
really did a lot to prove that entire relationship. And like I said, in the case of Kenya, the specialist that's going there is trying to find the alternate host for that rust on coffee, so that we can figure out what's the alternate host, what can we potentially eliminate in these other places that will help us fight the disease on the coffee plants, because really
we trying to train locals to look for that.
well, yeah, and at the moment, we just have no clue. We know that it infects coffee, and we don't know where it goes from there. So there's still lots to be done in the case of mycology in particular. So it would be great to train people, and in the case of Kathy, she definitely has trained people in Guyana. Actually, there's a recent graduate of hers came from Guyana, got her PhD now, and is now pretty much the state-sponsored mycologist of Guyana, because Kathy was able to go and teach these people how to find the specimens within their native environments, and they've continued to perpetuate that among their own students.
To me,
Yeah.
I noticed while I was going through the whole collection that, for example, there's a type specimen of rust collected by an Italian scientist in Ethiopia. And in the days of colonialism, country want to send scientists into other parts of the world, so to kind of say we're the European power that's dominant.
Yeah.
Or like Japan, we got a lot of specimens that was collected in Japan before World War II.
Yeah.
Foremost from Taiwan, from North Korea, which at that time was Japanese controlled area. So, and specifically, maybe she doesn't know, but Guyana, where she's collecting, is Venezuela claims that land. It's been disputed for hundreds. It's been in the news recently that because they found oil in parts of by Venezuela. It's the Essequibo region of Guyana. We say it's Guyana, but Venezuela said it's Venezuela. So that could be. And so Guyana wants to have scientists in this region collecting, looking for stuff, saying we found this in Guyana. So this strengthens our claim to this land.
Yeah.
But it within our lifetime, it could change. Who knows? Venezuela could take over if they want.
At the moment, the PUL is, we say that there are 110,000 specimens. Over 10,000 of these were donated by Eli Lilly a few years back, from their own various experiments and personal herbaria that they had. There are now 125,000 specimens within the PUR. So you can see Arthur had nothing to worry about. The Arthur collection is the largest of the two. And we have over 3,000 type specimens of rust fungi within that collection. So those are the specimens upon which a new species was described, the actual specimen that was used to describe the new species. The oldest specimen that we have in the collection is this barberry leaf from the first circumnavigation, collected during the first circumnavigation of the globe by Captain James Cook by Sir Joseph Banks. It is from 1769, it's collected in Tierra del Fuego, Argentina. And you can see this little tiny speck of dust on that leaf. And that leaf, that's probably a little bit bigger than life size of that leaf. And that is a rust fungus that was confirmed in the 1970s by Joe Hinnon. You can see the images on the side there, and where he found that the spores were still present on this specimen. It had been sitting in a cabinet for 200 years after it was collected. He found the spores. These are fungi that are obligate parasites on living plant material, so it wasn't sitting in this cabinet and grew moldy or anything like that. It actually was on the plant when it was collected in 1769. So when we have tour groups come through of new students, I often ask them, like, when was Purdue established? So this is 1869, 1769. This is 100 years older than the concept of Purdue University. Yeah.
Another couple of dumb questions. I'm
That's okay.
probably the only non-herbologist here.
I am not a botanist. I'm pretty sure they're the green ones.
Okay.
So good.
So is all the fungi in the collections, you know, the rust fungus and all that, is it technically alive if it still has spores in it?
They are most likely inert, but it's very, I would expect that they are not living.
But they have DNA
For shipping purposes, they are dead.
Yeah.
That's,
They have DNA you can extract.
we can extract DNA, and a lot of our studies that we've been doing have been focused on specifically on those type specimens, on getting the DNAs and putting those in repositories so that people around the world can compare their DNA signatures to those that we have as vouchered specimens.
But just the specimens actually die as opposed to go dormant for 100 years or so.
Yeah. The especially rusts are very difficult to get to try to inoculate back onto material after they've kind of become inviable after a certain amount of time. So yeah, these are biologically inert, but there definitely still is DNA present. It may be very degenerated, but it is still present in a lot of cases. And finally, we do also have several specimens from George Washington Carver and his studies in Iowa and Alabama of plants and rust fungi as well, among other things that, again, you can find on our website. Some of our more recent projects have been those conducted, like I said, a lot of work in Guyana, looking at fungal taxonomy, describing new species, these kind of really hard puffballs that are distributed by termites that were discovered, these plant pathogens of a greenheart is the plant, I believe. I can't remember exactly the name of the seed. Jeff is a recent PhD, now a postdoc that's did a lot with fungal taxonomy, both here in Indiana, but also looking at a lot of Central American countries. Paula got her project going, looking not at the rust fungi, not the plants, not the rusts, but at the insect parasites of the rust fungi. They're obligate parasites on the rusts, and she went through the collections and found that when a lot of these were preserved, at the time that they were preserved, there were larvae that were crawling on them and eating them. So these larvae were kind of serendipitously collected and preserved. And she went and did DNA analyses to determine how these larvae were dispersed, what their host range is, geographically, and also on the rusts themselves. And I like to put forth her case in particular because in the past year, there's been a lot of documentation about specifically the herbarium at Duke University being in line to be eventually closed. And it's, I believe, the second largest herbarium in the country. And there's a lot of people that are very upset about that. Not only the people at Duke University. And the idea that you can just like, okay, we can just ship things to other places and put our eggs in larger baskets. You know, who cares? We took a picture of it, we got some DNA sample, we're good. But in the case of Paula's work, this shows like, we never know the questions that we're gonna have 50 years from now, 100 years from now, whatever. So if somebody took a picture of this plant and this fungus 50 years ago, she wouldn't have been able to look at it for larvae and done DNA extractions on these specimens. So again, we don't know the questions that we're going to think of. So we need to have the physical specimens to help us provide those answers in the future. Yeah.
What's leading to the closure of the Duke herbarium?
Money. So the,
Or lack of one.
yeah, or just not wanting to spend it on that.
There's got a lot of money, they just don't wanna spend it on a herbarium.
Yeah, they don't want to rebuild a herbarium.
I don't understand the value of it.
Specifically with my own time here at Purdue University, again, I'll reiterate, I think people can vouch for me. I've only lived here 3 years. And during that time, for the past 2 years, since last September, before that, Dr. Aime has been away working for the National Science Foundation. So I've been really left to my own devices about figuring out what people are doing in the lab, the history of this place and everything like that. Thanks to a lot of great literature we have lying around the place. But since starting with the Aime lab, Kathy had written a grant to collaborate with people at the University of Kansas that were looking at plants from Africa, getting back to colonialism, plants from Africa that are held in North American area, and digitizing that information so we can make it available to the people that are in Africa that need to know what plants were available at the time, what plants still might be present, things like that. So for that project, we have been digitizing all the records that we have for not only the Creeple collection, the plants, but also in the Arthur collection, we have rust fungi on plants. We have additional plant information that we can provide from both of these collections. The REU, the research experience for undergraduates, that's a kind of an offshoot of our original grant that we're given by the NSF, has given money to help fund 2 current undergraduates. One previous person was Sydney Bunch. Sydney Bunch worked on the initial project. But I have 2 students now, Morgan Klein and Haymow, who will be co-authoring manuscripts on the rust fungi from African plants, because the people at the University of Kansas, I asked them, so we have all this plant data, we have all this fungal data that came along with the plant data. What do you want us to do with that? And they said, we don't care. So we're going to be publishing that list to make that available to people worldwide. Most likely the proceedings of the Indiana Academy of Sciences within the next year, I hope. So now it's where I go off on a tangent and talk about myself for a bit. So why am I here? I've already said I'm not a botanist. Pretty sure they're the green ones for the most part. I'm not a basidio mycologist. I don't work on mushrooms and like fleshy big things. I work on the little guys. My history has been looking at the Ophiocordyceps, which are the zombie ant fungi that you think of. This is a new species that we described from the European red fire ant that was invasive in Acadia National Park in Maine. I got my master's and PhDs at the University of Maine in Orono. I then, after Maine, went to Florida to study ambrosia beetles, which are beetles that carry fungi in various parts of their body to one tree to the next to start these fungal gardens, so that they don't actually eat the trees that they grow in. They just grow into the trees, then they start growing these fungi onto the substrates. And that can be fine for the beetles, good for the fungi, good or bad for the plant, depending on the type of fungal infection. But I worked on those for a couple of years while I was in Florida. But my background before both of those, the ways that I got my master's and PhD, were looking at these zoosporic eufungi while I was doing my degrees at the University of Maine. These things that have flagellated zoospore stages, kind of like sperm cells in humans. You have to remember, fungi are more closely related to us than they are to plants. So we have a common ancestor where we are modal. These things were modal. In the case of mushrooms, they lost that motility. And right before coming here 3 years ago, I was at the University of Michigan for 5 years, where we established the collection of zoosporic fungi at the University of Michigan, or the C. So now you're probably thinking exactly what is a zoosporic fungus? I thought the same thing. I thought after this is my son Jasper. at the time, he was 7 months old. This was, he's now 2.5. This was actually taken February 14th, 2 years ago. I remember. And I thought, okay, he's 7 months old. He needs to know what fungi are. So we got out our first book of microbes, and you can see viruses, bacteria, fungi, and more. So there's going to be fungi information here. It's going to be valid and good. In the second the fungal page, it said that fungi have no cilia, small hairs, or flagella and cannot move around by themselves. And that is the sole type of fungus that I worked on for my master's and PhDs. And the thing that really got me were that the authors used all the right terminology. So they didn't Google anything to prove themselves right or wrong. They just heard this once and thought that's true. That's a true statement. So I did what any sensible parent would do. I went on Twitter and I contacted the authors and said, actually, if you look at our recent publication in PNAS, which made the cover photo of that issue, you can see that there are many characteristics of the fungi, including that they have flagellated life stages. That tweet at the time reached an audience of 17 views, not 17 likes.
Wow.
So it's always nice to get out there and talk about these things a little bit more.
Yeah, good.
Yeah, get
And
more
was
people.
their response something like, don't confuse us with the facts?
They never responded. But before I came here to work on the rust fungi and be associated with the herbaria, I knew Purdue University because of John S. Carling. John Carling was a graduate of Columbia University who started working on algal organisms, but he kept being hampered by these chytrid zoosporic eufungi organisms that were infecting his algal plates. We colloquially now call these chytrids. There is now the Chytridiomycota, but there are still other branches of the fungal tree of life that sometimes we call chytrids, but they technically aren't chytrids. But I may use those term chytrids and zoosporic eufungi interchangeably. Carling was the head of biological sciences here at Purdue.
Yeah. What's a chytrid exactly? Is it a type of fungi that's maybe stable and rooted for a while?
It is a true fungus. It is related to mushrooms and everything else. It has this zoosporic stage at one point. It's a single cell with a posterior flagellum, like I said, much like kind of a human sperm cell. We found actually very recently it's probably diploid at this stage, which in not just haploid. And it will find a new substrate. It will insist producing that cell wall that characterizes most fungi. Up until that point, though, it is only bound by a plasma membrane. It's not bound by a cell wall at that point. It forms a cell wall. It will then somehow expand or send out rhizoid structures to increase its surface area and its just overall body or thallus. And somewhere on that thallus, it will produce a new zoosporangium or multiple zoosporangia where more zoospores are produced and the whole process starts over again. It's for the most part, they're an asexual group.
Yeah, because it's different.
Yeah,
Okay.
but they are kind of single-celled organisms.
So they're not basidiomycetes, right?
No, they're within their own phylum. They are the Ascomycota and the Basidiomycota that most people think of when they think of the fungi, the Dicaria. They are they always put them at the top of the page, and the chytrids are way down here, but they still are within the fungal kingdom with a capital F.
I don't know how to pronounce it. Mycota.
So the Oomycota are stromatophiles that are elsewhere on the tree of life. They're just a convergent form of fun. So the oomycetes are fungi with little f. Chytrids are fungi with big F.
Do they typically grow on algae? Is that their host?
They are everywhere. And including, I'll tell a good story about where you can find them very, very easily. So the chytrids, Carling started working on the chytrids. He came to Purdue University in 1948. He was the head of the biology department for 10 years. He told the president at the time of his hiring, he will only be the department chair for 10 years. They pursued him for many years to come here and be the department chair. He actually helped design Lilly Hall. In 1958, when he reached that 10-year deadline, he called, I think President Huffdy at that point?
Yeah.
he called him and said, okay, 10 years are done. They said, no. We will make you a specific distinguished professor, to the first John S. Wright Distinguished Professor, so that we can keep you here at Purdue for as long as possible. And eventually, I believe his lab closed in 1985. But it's been said of him that he was responsible for reshaping and redirecting the biological efforts at Purdue. He was, again, another one of these world-traveling mycologists, though he started from humble beginnings as a White Sox reject. He didn't make the team. He got lost in Belize, I believe, and accidentally rediscovered some Maya cities while he was working for a company on how to mass produce chicle. And he had a rivalry with President Dwight D. Eisenhower, who was president of Columbia University when he left. And eventually, Carling would work on the presidential yacht that was decommissioned by Eisenhower to then become an NSF vessel. And he did some work on it in India. He literally wrote the book. I keep saying everybody writes the book, but he did also write the book on chytrid fungi, one of the books on chytrid fungi. And his specifically is very beautiful. Every other page are these illustrations that he did of these organisms to make them kind of understandable to people that don't have a relationship with him or just didn't have access to microscopes. And again, I have a full biography written about John Carling. He is widely known within the Mycological Society of America, specifically because the presidential address, the keynote address of the MSA meetings is known as the Carling Lecture. It was because in 1995, after his passing, he gave a donation of $10,000 to help perpetuate that lecture series, and it was then named after him. So you go to MSA meetings and people talk about, oh, who's speaking in the Carling Lecture? Make sure you make it to the Carling Lecture. And you say, who's Carling? And they're like, I don't know, some dead guy probably.
What position did he play? What position?
I don't know. That's a good question. I don't know.
Did you marry somebody
I don't-
from West Lafayette?
So yeah, so he married Sarah Christian. Paige Burrell Johnson. I think she was actually from. I think they met in Connecticut, but I can't remember. Her family's on vacation or something. But she continued living here. She passed away in 2007. I was curious if anyone here might have known her. Carling passed away in 1995. What?
I did know. I think she was a quilter.
Okay,
Well, let's see if I can figure it out.
I'd love to know more. If you can find any information, please pass it along.
We have some sewing friends Tuesday, so maybe between us we can figure it out.
Great.
Is that his daughter?
So yeah, that's his daughter, Sarah. Sarah did attend West Lafayette High School. I believe this is her high school photograph. I think from what I was able to find out, I think she was 16 in this photo. And she married. She went out to the University of Colorado, I believe, is where she got her degree. And she married a man, moved to Chicago, and passed away before the age of 30. And I'm not sure exactly what the circumstances about her. Yeah.
So chytrid, is that how you pronounce it?
Yes.
Are they collectible and you put them in a fungarium, or do we have any specimens? of it?
We have, so Carling wrote one book specifically about Synchytrium. Synchytrium is a chytrid pathogen on plant material. And we have some chytrids. There's some chytrids at other places. I'll talk about here in a second. But we do have some in our collection, but not that many. But what we have are on dried plant material.
Yeah.
So it's completely invaluable. Once these things are desiccated, some of them have resting structures. And one of the people that also studied them, I've seen some handwritten notes from him saying, in the hands of someone who precisely knows exactly what they're doing, maybe you could revitalize these from these tissues that are preserved on slides. But
You still have DNA.
they still do have DNA, definitely. And I would love to do an experiment specifically with the Synchytriums that we have from Carling, because Synchytrium, there's not that much known about Synchytrium, and at this point, that particular genus is recognized as being within its own order, the Synchytriales. So I'd really love to do more about the biodiversity of the Synchytriales from what we have here.
Inception, man. Is there any specimens in PUL or PUR that he collected?
Not really. A lot of his materials were donated to Miami University. Carling inspired many people to continue working on chytrids, and among them was Joyce Longcore. Joyce Longcore worked with Fred Sparrow, seen here and here. Fred Sparrow, always dapper, hilariously so. Would always, I've heard stories of him going like on collecting tours in bogs, wearing like seersucker suits, and getting mud on his shoes. But Sparrow wrote the other book on chytrid fungi. It's literally about this thick. Sparrow liked to talk, Carling liked to draw. But their two works complement each other very well. But Joyce Longcore was a student of Sparrow at the University of Michigan. She herself grew up in South Haven, and she started working with Sparrow in the 1960s. Would eventually, during her undergraduate, she would marry. She went to IU to complete a master's program, but then she kind of would say that she became a housewife. She followed her husband upon his career trajectory. He went to work for the USGS. She worked at Dow Chemical, I think, in Maryland at the same time, and eventually she would end up at the University of Michigan. The University of Maine, where she would fall back into a relationship with her best friend from college, Alma Homola. Alma Homola had married Richard Homola, and Richard Homola was a fungal specialist who had gotten his degree from another fungal specialist at the University of Michigan, aside from Sparrow, or actually had gotten his degree from Sparrow as well. Or Sparrow was one of the people on his committee member. And while Joyce was at the University of Maine, she started getting back into the chytrid fungi. And specifically, in the late 90s, there was an outbreak of deaths of these blue poison dart frogs in the National Zoo in Washington, D.C. And they didn't know what was causing the disease. So the people in Washington, D.C. reached out to Donald Barr, and Donald Barr was a specialist on protozoa, who did a lot of work with chytrids in the 1960s and 70s and 80s, actually named the first order for a long time of chytrids, Spizellomycetales, for which he named the Spizellomycetes for Sparrow. It was in honor of Sparrow, Spizello. And they reached out to Barr. Barr was a Canadian citizen, and these were problems at the U.S. Zoo in Washington, D.C. He didn't really want to get tangled in political problems, and technically he was supposed to be working for the Canadian government, but he referred them to this woman that he had just recently trained on zoosporic electron microscopy, that had been interested in it, Joyce Longcore. Joyce Longcore would obtain some collections and specimens from these frogs and found these little circles, these little spheres in the frog's skin. And occasionally there was this septum, this wall structure between the circles. And she, from this, was able to isolate using her standard chytrid isolating techniques. She was able to isolate from these skin cells the first pathogen of chytrid fungi on a vertebrate host. At the same time that she got this pure culture, people were seeing in wild populations on the other side of the planet in Australia that there was a very similar thing happening to their frog populations that had these same looking structures in the skin of the organism. And they kind of jumped the gun and said, we're going to call this chytridiomycosis because we're pretty sure chytrid does it, but we didn't isolate it. But Joyce had isolated it, and she named it Batrachochytrium dendrobatidis, or BD for short. And like I said, this is the first chytrid that was found to be a parasite on amphibians. And it's become quite the thing for Joyce, who is a self-described granola. She's a wallflower. She doesn't like to talk in public. She grows half of what she eats, and her husband kills the other half. She is a Mainer through and through, or a Michigander who now lives in Maine. But BD has become a really big deal. BD is we now know is chytridiomycosis of keratinized tissue. It leads to disruption of osmoregulation. Kind of it infects the keratinized tissue of the frogs. So in the case of tadpoles, it's going to mostly be mouth parts. In the case of the adult frogs, it could be mostly like the drink patch areas and between their toes. But it eventually just leads to this osmoregulation disruption. And they kind of just slowly have little tiny heart attacks. And it has been correlated to population declines in over 500 species of frogs worldwide, and probably quite a number of extinction events. And it hasn't just been around since the 1999 when it was described. This has been around for quite some time. Just there were only 5 people in the world that happened to be studying chytrid fungi, and they weren't looking at frogs because it didn't grow on frogs. We never thought about frogs before. But it's since been a really big deal. There's actually another species, B. salamander vorans, which we don't call BS because we can't say BS at conferences, but we can say B. sal. So we call it B. sal. But there's B. sal and BD. B. sal has so far been relatively contained in Europe and Asia, but it really, the reasons behind the spread of BD are numerous. May have something to do with pregnancy tests in the 1950s and 60s. But it's believed that when it was discovered that salamander vorans existed, salamander trade around the world was effectively like 99% shut down. We were trying to keep this stuff contained in these locations. But most recently for BD, there was a paper that came out in 2022 that correlated BD population and infection on frogs within a die-off of frogs, which then led to an uptick of mosquitoes, which were infected with malaria, that then gave humans malaria. So this little microscopic organism that nobody knew about 25 years ago has human health implications. So it's a really interesting study focused on Costa Rica and Panama that shows this interaction in its natural environment.
So that B. sal affects amphibians too, is that right?
It's salamanders almost exclusively.
Okay,
Yeah.
and the other.
BD is wide ranging. It infects a lot of stuff.
Okay.
It's just amphibians in general.
Toads too?
The Australians would love for it to kill cane frog,
Yeah.
cane toads, but it doesn't. It's just anything with a nice aquatic stage.
Yeah.
It actually bullfrogs don't really die off from it, but they're really good reservoir species. They can just accumulate so much. So then you send you find a bullfrog, and it goes to a bullfrog farm where people actually consume bullfrogs, and it can wipe out farms. There was a example where someone at Michigan went to the next town over. They went to their like Asian food market, bought some bullfrogs there that had actually been grown in Brazil, and they were able to isolate chytrids from that leg from Brazil, or from that market in Ann Arbor, and genetically it was identical to the stuff that they had found in Brazil in wild populations. So human commerce, man, we spread stuff around like crazy. But the description of BD led to this renaissance again to zoosporic eufungi, and has led to a lot of interdisciplinary collaborations. The public just becoming more aware of these things. There are children's books about chytrids. There are video games about chytrids. They do chytrid specials on PBS and MPBN. This is a album cover of an album that just came out last year from British death metal band Slime Lord. The name of the album is Chytridiomycosis Relinquished. So it's reached quite different levels of humanity at this point. I personally was trained from an NSF Partnership for Enhancing Expertise in Taxonomy grant, a PEAT grant, through Joyce Longcourt at the University of Maine. And I collaborated a lot with Martha Powell during that time. Martha Powell was the department chair of both JMU and University of Alabama in the 90s and 2000s. And specifically her connection back to Purdue was that she was a student, a postdoc here on 1974 to 1976 with Charles Bracker, who himself was the George Cummins Distinguished Professor in 1993. So going back again to Cummins and the Arthur collections. It's fun. The mycological world is somewhat incestuous. We all somehow intertangle back to each other. Finally, getting kind of, I hope that you can see, I really enjoy this stuff. and I think if anything has prepared me for public relations, it would be studying chytrids and getting people interested in these things that are, by definition, that for past experience, they haven't gotten that much attention. So I love to do outreach opportunities. And like I said earlier, we have been at Springfest past couple of years. This was 2 years ago when we had a specimen mounting event that we're going to have actually outside this year. So hopefully we'll get some more people, but the herbarium itself or the herbaria themselves will be open. So you can come down to Lily G447 and come see me. I'll probably be there all day long. National Geographic came out in April this past year. That is the first time that fungi have ever been featured on the cover of National Geographic in the 130 years of National Geographic. And I believe I'm quoted 3 times in that. So I finally made my grandfather proud. I've gone out to Remington Carpenter Library and talked about, they wanted to just talk about fungi and how to grow fungi. So I provided some samples of materials that we had in our collections and also some of the literature that we had. We've hosted various groups. I believe this was a Midwest chapter of the American Phytopathological Society that came on campus to visit our reading room and some of the specimens that we had. I'll go to various other institutions to talk about our collections and also the chytrid fungi. I'll go out and set up a thing in the middle of the forest just to talk about our collections. This was actually, I believe, the Fall into Nature event last a couple of years ago, and I just today received a notification about the Spring into Nature event at the Hort Park here on campus. And finally, the thing from the exponent there in the middle, that is a letter to the editor that I didn't mean to be a letter to the editor, but the exponent ran an article about, a cover article about the importance of cursive handwriting. And specifically for curation and for looking at all these old records, there's a lot of cursive and it's various degrees of legibility. So when they wrote this thing about how students were coming into college and didn't know cursive and couldn't write cursive and couldn't read cursive, and I sent this letter to the author saying, thank you for bringing this to people's attention. You know, I struggled through this a lot, and when I have a student, traditionally when we have a new specimen, we put a stamp on it, we put a number on it. Right in the middle at the bottom, we put what state or country it's from, and then over here we write the name of potentially the fungus, and then the name of the plant that it's on. And historically, that's been done in cursive. So we continue to do it in cursive. So when I had one of my African project students, and I said, okay, so then I want you to write this. And she said, no, I can't do that. I don't know how to do that. So I wrote this letter saying, you know, this is very important, and I hope people do actually take that in consideration and like try to add this very simple tool that I learned at the age of 7 into their scholastic toolbox. They asked if they could publish it, and it kind of got postponed a couple of times and eventually made it into the issue of the exponent that was shipped out to new students to their homes. So hopefully some parents might have seen this and like gotten like, hey, well, it's too late for this one, but this one's younger and maybe we can train them cursive. But also my letter there was seen by the Purdue Alumnus magazine, and they had just done 150 years of Purdue and 150 people and 150 places, 150 things. And they emailed me and said, we completely skipped over the Herbaria, J.C. Arthur, George Cummins, any of these people. They weren't on any of our lists. So kind of as an apology piece, they wrote this excellent article about us in the Purdue alumnus in this past spring, this past year. And I would really encourage you to check that out because they make us look really good. We're actually going to be doing another version of this very soon through Purdue Ag Communications. They're going to be writing a very similar stories, and they're going to put me on camera. And as you can tell, I've already had a meeting with them. It went over. I don't know why. So they're going to try to film me for like 3 hours and see what kind of story they can salvage for that.
Is-
Yeah.
So we can say that Purdue's herbaria are functional, right?
Definitely.
What about IU and what about Notre Dame?
I don't work there.
I know, but are they doing anything that you know of?
I
You haven't heard?
I don't have personal experience with those.
I used going, still there,
Yeah, but they've put it away, haven't they?
I was gonna say,
well, it's in a room like the herbarium. There's, Rich Hall is the guy that, knows, is
Right.
done most of the work with him. You got Rich?
I met Rich last year at IAS, but
Yeah, he'll be at
the former person there.
They got rid of it. Yeah, it's
Yeah.
terrible, you know,
Just this
I
past
use
year.
as bad as Purdue in a lot of ways, you know, like, they, what's his name? The former,
I can't remember it either, but just this past year, he was
and-
shown out.
yeah, it was, he'd been there and,
So yeah,
he
Rich
was a
is,
lobelia expert.
yeah, so Rich is in charge. So I know that it's still there and but I don't know who. Well, we still issue loans. We take in materials. I just like I was saying earlier from my interactions at the Indiana Academy of Sciences meeting last year. We've accessioned just from one group, we've accessioned 150 specimens of fungi. And like I said, Kathy's still traveling. We're sending out materials. We're accepting materials on loan so we can do our own studies and send those back.
Yeah, there's no question.
Yeah.
I mean, to think how close we came to probably shutting down this herbarium.
Sure.
Here,
Yeah.
if they hadn't hired Nick to start digitizing things, I mean, we would be in a world of hurt. It
Well,
would
who
just be
knows?
shut away.
That's very possible.
So yeah, we're continuing those efforts and still also as the curator of fungi, I'm also in charge of the living culture collection that we have in the Aime lab, which is another 8,000 things that I have to think of. So there's always something to do. It's a certain degree of job security. But it's also like stuff to get stuff accomplished, but there's always something to do.
Is that guy, Jin Rong Zhu, is he still working at Purdue?
I don't know that
You
name.
know Jin Rong?
Oh, yeah, well, yeah. Mycology. Yeah. He teaches general mycology.
Yeah, he's pretty good.
So-
About Notre Dame, I think it's really the largest herbarium in the state. And I think it's kind of, it seems to me it's like unused, or I mean, it's like kind of like one of these things. Looks like herbaria everywhere almost. There's like, it's not.
We have quiet days and then we have days where we're really busy.
Oh, yeah, I call myself the Maytag repairman, you know.
Yeah.
The oldest guy in town.
Yeah.
Nobody can, but I know Scott Nemesnik goes to the Notre Dame Herbarium to do stuff too, so he's told me he's been, so it's accessible to some people, so.
Okay. I know Butler just redesigned theirs and it's really nice.
Oh, really?
Yeah.
That's good.
Yeah.
Butler should have they usually have an open house in the herbarium every spring, so it'll be maybe you should go.
Well, we had that one thing last year at the IAS, or like the week after the IAS, they had an open house and
You know that?
yeah.
Oh, that's where I saw you.
Yeah. But I guess that's all I've got to say.
I gotta say, I hate to be critical, but this was a pretty spore presentation. But you're a fun guy.
I say I purposely pronounce it fungi.
Yeah.
Well, I've been known to mispronounce words all my life.
That's okay. So I thank you.
Yeah, but it was interesting. Am I the only one in here that came in here not knowing anything about? There's been some pretty intelligent, you know.
Well,
But we worked in it, you know, so.
Yeah.
That's all right. Not
I'm
everybody can.
upset and it's not all, we're the only ones, there should be like, 300 people here.
I guess.
Yeah, I was surprised there
Okay.
weren't more folks.
I should have said thank you to all the online people too, but I know there's a couple still here. But
But if you think fungi aren't appreciated, my specialty was algae. Now trying to describe that to people.
yeah, I know.
What are they?